Okay, so we start now. So now we are talking about another class, superfamily of proteins, which are the regulators of many signaling pathways. So that's why we're saying regulating the regulators, because one G protein can regulate several G proteins, right? So the difference between kinases and G proteins are that kinases phosphorylate a protein and that turns on a signaling pathway, right? It can inhibit a signaling pathway, it can turn on a signaling pathways. But G proteins are different in a way that they act as molecular switches. So suppose a signal is coming from the top, and if this signal is off, the signal stops here. It will not proceed further. Only when it is on, it will go down there. So they are molecular switches, like on, off kind of thing. Okay. So, and they are called G proteins. They are also called GTPases. Both are the same thing. So G proteins, they are called G proteins because they either bind GTP or GDP. If they are in the GTP bound form, they are on. If they are in the GDP form, they are off. So this is on, this is off. Okay. Now G proteins can spontaneously hydrolyze to GDP and turn off, or there are these special proteins called GAP, GTPase activating protein. They can also hydrolyze GTP to GDP and then it is turned off. So if you delete GAP, obviously it will be in the on state for a very long time because the hydrolyzing proteins are missing. Is it making sense? Now, as a biologist, if I ask you that, so imagine this is G protein and this is GDP or GTP bound. Let's say this is GDP. So do you think this GDP can get converted? Hydrolysis is easy. Phosphate can get hydrolyzed. But you cannot add a phosphate while GDP is bound here. So you have to remove GDP and then you have to bind GTP to make it active again. So the proteins which do that, they are called GEF, G protein exchange factors. So they exchange GDP for GTP. Makes sense? So if you don't have GEFs, then they will stay in the offside. So initially it was John Kurian who solved the structure of how GDP is released and how GTP binds. He's a very famous crystal structure person. So it's like GDP is bound very tightly. So these GEFs, they open up the thing so that GDP leaves and then GTP comes and bind and then it is tight again. Makes sense? Okay. So so once again, when they are bound to GTP, they are active, they are on switch, downstream signaling occurs. When in the GDP form, they are off. GAPs hydrolyze it, GEF exchange, they remove GDP and they put GTP back. Okay. So why I call G proteins as master regulators? Because G proteins, a single activated G protein can activate hundreds of other G proteins. So they are master regulator. And now we have to regulate the master regulator because G proteins play a very important role in cancer. Okay. So there are two. So G proteins can be classified into two categories: heterotrimeric G proteins and small G proteins. Okay. So heterotrimeric G proteins, so this is a receptor, this is heterotrimeric. Okay, spelling is wrong here. Heterotrimeric, so there are 7 receptors. Receptor is not a G protein. This is just a receptor, and see, this is an extracellular domain, then this is the domain which is embedded in the membrane, and below, can you see this thing here? This is the lower, this is in the intracellular part. G proteins are attached there. Okay. So these are called heterotrimeric because there are 3 of them: G alpha, G beta, gamma. Beta, gamma are joined together, G alpha is separate. So GDP or GTP binds to G alpha. If it is in G bound state, it is active. GTP bound state, in GDP bound state, it is inactive. So what happens when a ligand comes and binds here? GDP is exchanged for GTP. When it is in the GTP form, alpha dissociates with beta gamma, alpha will turn on its own signaling, beta gamma will turn on its own signaling. Is it making sense? So you have the receptor. So receptor, let's say this is the membrane. So receptor is there and it has a tail. Okay. On this tail are attached this heterotrimeric protein G alpha beta gamma. Beta gamma are together, alpha is separate, but they are all together. Okay, they are all together in the inactive form. GDP is bound. At any given state, it will be not without GTP or GTP. Something has to be bound. They are never without that, okay, because their binding is very tight. So it is at the GDP bound stage. Now a ligand comes from outside, binds here. So the tail changes conformation. Because of the binding up here, the tail changes conformation. This is this tail, the black tail. And because these proteins are bound to the tail, now they will undergo conformational change. GTP will be replaced with GTP, and this will turn on its own signaling. Beta gamma will dissociate, they will form their own signaling. Make sense? So that is, so there are many heterotrimeric G proteins. Then comes the small GTPases. They don't have this any membrane receptor coupling or anything. They are just small proteins which are either bound to GTP or GTP. Okay, and the most famous protein in this small GTPase family is RAS protein. And RAS is one single gene which is responsible for maximum number of human cancers. Okay, again the same on-off switch. Okay, so this is heterotrimeric G protein. So a ligand comes and binds here, and once it binds, then this exchanges GDP to GTP, turn on downstream signaling, beta gamma turn on their own downstream signaling. Now let's look at this part here. This is where we will focus most of our attention. So these are small G proteins, and small G proteins are further divided into many family members: RAS, Rho, RAB, ARF, and RAN. Okay, then all of these have many family members. So RAS, these are some members. They are involved in cell proliferation. Rho is involved in migration. This is vesicle transport. ARF in vesicle formation. So you can see RAS and Rho, they are directly involved in migration. Migration means metastasis. Proliferation means uncontrolled cell death. So these are most important in cancer, RAS and Rho, right? So look at the members. So this is a little bit, you have to pay attention. Although the family is called RAS family, right? This small GTPase family is called RAS family, but within RAS there are so many other proteins there. So RAS family members have 36 members, including RAS. Rho has 20 members, RAB has 61, ARF has 27, RAN has 1. Okay, and then there are different kinds of functions here. Okay, so let's look at Rho family first, because we will focus mainly on RAS, so I'm telling you about Rho first. So this is normal circumstances. There is a balance between GDP and GTP, and GAP hydrolyzes to this, JEF brings it back. This is downstream signaling. Look at this thing. This is regular thing. But when this is broken, so now it cannot hydrolyze, it will remain active for a long time, right? Look at the amount of signaling increased tremendously, right? Or if this is working overtime, it doesn't let GDP stand there, it immediately converts to GTP. That also will increase downstream signaling. So all of these things will lead to cancer. Okay, so RAC1, another family member, this is also involved in angiogenesis, invasion, everything. Okay, another ones, RAC, Rho, these are just different kinds of mutations which occur in different cancer, and that also leads to cancer, right? So these mutations, what they do, if you have to keep these GTPase active all the time, what should these mutations do? Tell me. Histones. So that is increasing the expression, but increasing the expression is not the main thing here. It is there to some extent, but if it is undergoing mutation, what should happen due to the mutation? Can you tell me? Yes, so it will keep it bound to the GTP bound state. All these mutations have one purpose: keep it in the on state, keep it in the GTP bound state. Okay, so now we'll come to the biggest culprit of the G protein family, which is RAS. So look at this single RAS activating so many pathways. Remember, earlier today I showed you VRAF and how good it was, Plexicon compound. RAF is just one of the guys here, turning on only this signaling pathway. But if you inhibit RAS, you turn on everything at once. It's a master regulator, right? On top of the things. All the growth factor signaling comes to RAS. RAS is the one decides it can go down or it cannot go down. So you can see how important it is. Yes?
Would this come under the category of oncogenic addiction?
Oh, absolutely.
But it's naturally this way, right?
No, so good question. So what happens is that RAS is supposed to be activated only when growth factor signaling is occurring, right? And then when the growth factor signaling is not come, this is turned off. But if there is a mutation which makes it independent of the growth factor signaling, it's on all the time. We saw such, remember I showed you the PET scan when you inhibited RAF. So you have inhibited one guy and that cleaned up the signal so much. So just imagine if you inhibit RAS. If RAS is the culprit, you can only inhibit it if it is the culprit. But you can imagine how bad it is if it is the culprit, right? Because it will activate so many pathways. So this is what it is saying. This is answering your question. RAS, under normal circumstances, it will respond to extracellular stimulus. And then only it will be activated, right? And RAF is down here. But if suppose gaps are missing or RAS is mutated, that's always in the GTP bound state, it becomes independent, and that's really bad. Okay, so now let's look at RAS family. So now we are focusing on RAS. So we started with the whole super family of G proteins, 2 heterotrimeric small G proteins. Then we saw small G proteins are also many kinds. We looked at Rho protein mainly. Now we look at RAS, and the rest of the talk is all about RAS. Okay, so there are 3 kinds of RAS: HRAS, KRAS, and NRAS. Okay, these are slightly different sequences. KRAS is the biggest culprit and the hardest to target. So all of these 3 RAS, they are activate MAPK pathway, PI3K pathway, RALJF. Don't worry about these pathways because this is very complicated. Basically, they turn on many signaling pathways. Okay, and then you can see that, so now look at this thing here. KRAS, it is mutated at this site, 12 and 13 site. It's a glycine, and this mutation locks it in the GTP bound state, so it's super active. Okay, and look at it, how many different kinds of cancer. Remember I told you pancreatic cancer is the worst cancer because it's diagnosed at stage 4. There is no drug for it. RAS is the main problem in pancreatic cancer. Okay, so you know, 12 years ago, it was up until 12 years ago, it was 100% known that nobody can target G proteins because their binding to GTP is extremely tight. Okay, so nobody could ever inhibit any G protein, RAS or anything else, despite it was such an important target. So people used to inhibit the receptor signaling, or they would inhibit RAF, all the downstream guys or all the upstream guys, but they could never target G protein, including RAS. So 2013, everything changed completely, and I'll tell you about that, what changed. Now we have direct RAS inhibitors, and we can inhibit the RAS signaling, and you can imagine how big of a finding it is. Okay, so this is just to show you. Somebody was asking me yesterday, what is a proto-oncogene and an oncogene? Proto-oncogene is like RAS. Under normal circumstances, it gets a signal from upstream and it works. But RAS, if deregulated, can become an oncogene. It has that potential. So proto-oncogene is not bad, but not all protein, if you make it deregulated, will cause cancer. So they are not proto-oncogenes, but RAS is one of them. Okay, so it becomes a proto-oncogene. So like you guys were saying, there are many ways G proteins get deregulated. Most common is direct mutation for RAS, for G proteins. Okay, overexpression of JEF. JEFs are those which, what do the JEFs do? Yes, so if they are overexpressed, they will always keep it in the active form. Loss of negative regulators, GAP hydrolyze it. If GAP is gone, they will never hydrolyze to GDP. Remember, you remember gap is the one which converts GDP to GDP. So if they are gone, see, gap is the one which is converting to GDP. If that is gone, there will be no switch off mechanism. Overexpression always leads to problem. Now if you keep it active by different kinds of mutation, different kinds of post-translational modification, that also will give rise to disease. Sometimes you have a spliced form which is always active, that is also cancer. Okay, so this is just to remind you that under normal circumstances, receptor tyrosine signaling gives signal to RAS, which is inactive, but only once the signal comes it becomes active. This is normal circumstances. So wild type, there is a balance between exchange of GDP to GDP and hydrolysis of GDP to GDP. But when this mutation occurs, hydrolysis is impaired. It's only exchange, so it's always keep it active in this form. So if you can inhibit it here, it will completely switch off that. Make sense? You guys are with me? Okay, now see, RAS alone. Remember the three kinds of RAS. So among the three kinds of RAS, if you combine all the mutations, most of it belongs to KRAS. KRAS has 86% mutation in cancer, HRAS 12%, NRAS 3%. And look at the mutation. Most of the time it is the G12. G12, one site is mutated and it locks it in the GTP form. Okay, here also most of them, here it is G12, but it is also Q61, G12, G13 in different kinds of cancer. HRAS, NRAS not so bad here. Okay, so this is, look at the thing. These are all the mutations only in RAS protein. If you just only look at RAS protein, look at the contribution of HRAS, KRAS and NRAS. 83% KRAS, 13% NRAS in all cancers. Here, different cancers, look at it. So usually KRAS is the biggest culprit here. Okay, this is telling you all different kinds of cancer. Look at the green one. G12 is the biggest issue here. G12 is the one, and G12 green is KRAS. So now you guys get an idea that mutation is the biggest problem, and that too at G12. Okay, so this is RAS subtypes in cancer. Okay, you can see, for example, melanoma, mainly it is NRAS. Okay, melanoma is what kind of cancer? Skin cancer, right. Okay, so KRAS mutants in lung cancer, it's all G12 mutations. Okay, so RAS is the biggest, if you have to name one oncogene which is the biggest culprit in human cancer, it is RAS. And RAS was undruggable, so just imagine how bad it was. Okay, so this is just to show you RAS family of proteins. They give rise to cell survival, cell proliferation. They change the microenvironment, tumor microenvironment. So they are extremely bad things. So why this is a big deal to target GTPase is historically undruggable. Up until 2013, they were undruggable. Recent breakthrough. Okay, this is I will tell you that. Okay, so why it is such a big deal? Okay, so before RAS direct inhibitors were discovered, so RAS is a membrane bound protein. Okay, so just like that, membrane bound. If this is the plasma membrane, so there is one thing about RAS. If you don't let RAS go to the plasma membrane, it is ineffective. So when I joined my university, there were many scientists who were just working on those inhibitors which would not allow RAS to go to the membrane. And they were considered very important because they could not target RAS. Okay, so people were targeting downstream proteins, they were targeting upstream proteins because there were no direct RAS inhibitors. Now they are direct RAS inhibitors. Okay, so now what we do, that RAS is going from GDP to GTP state, like cycling. So when it is bound to the inactive form versus active form, so when it is bound to inactive form, and then active form. So when it is in G12 stage, which is the biggest mutation there, okay, this G12 thing, now we have made covalent inhibitors of RAS G12C. There are many, many of them, and once you bind them, and if you do the PROTAC also, you can even degrade it. So you can directly inhibit RAS, and if you make a PROTAC version, you inhibit it and you degrade it, okay? But how did we come to this? So this was the first compound, okay, in 2013, these two. These were the first compounds and the first time ever RAS was inhibited. And this was discovered in my postdoc advisor's lab. And this is one of the biggest discovery of this century, okay, one of the biggest discovery of this century. I mean, the field is really indebted to him for this discovery. So what he found is that this is GTP binding site, right? So he found that there is an allosteric site within that, and his students discovered that pocket, and you know, he's a chemical biologist, so he made the compound which targets that allosteric bind, and that is what is compound 6 here. And once his lab, he opened up a company and everything, and once his lab discovered, the whole world started making it. So all of these compounds, they are in clinical trial, they were all after him, okay? So that was, so what he found is that, so this is switch 1 and switch 2. Don't worry about what are switch 1, switch 2. So I did not tell you about in details here. See, this is switch 1, switch 2. So what happens is that when it goes from GDP to GTP side, this switch 1, switch 2 is the one which are moving. Okay, so these are the ones which are binding GTP, GTP. Okay, so he found that when it switches from one to the other state, a new pocket is formed. It's not there all the time. When it is switching, that's when it is formed. And he targeted that pocket. Okay, so that's what it is shown here. That look at switch 1, switch 2 here. So see, look at this yellow part here. So this is on stage, this is off stage. Can you see they are different? Can you see that they are different? Right? Switch 1, switch 2. So when it is in this off form, a new pocket is formed which is absent here. So he made a compound which can target RAS in the off stage. This pocket is only created when it is in the off state. This is what kind of pocket? Can you tell me? It is allosteric, but a pocket which appears only. What is that word? Shivani? No, myristoylic site is a special site in BCR-ABL, but what is the other thing? Cryptic pocket in one site only, right? It is obviously an allosteric site. See, this is the lipophilic pocket which appears there. Okay, so that is what was, so now there are many compounds, and this is a covalent inhibitor. This is a this is a acryl group. So as such attacks here, it becomes a covalent thing. Okay, so this is where the compound is bound here. Now you can see. Now there are so many compounds people have formed. Look at this. This is allosteric site. This is GDP site. This is competitive site. This is binding to the allosteric site. You can see that, right? So this is between the switch 1, switch 2. Okay, and this is the switch 2 pocket which creates, and that's where the compound binds. So once that pocket was found, now there worldwide people are making many compounds targeting them, and they are all in this thing. So before, like see, so many inhibitors are there, okay, for RAS now. These are direct. Before this, people were going upstream and downstream to inhibit RAS signaling because nobody could directly inhibit RAS. So this is my advisor's name, Shokat. So Shokat lab identified the first KRAS inhibitor, okay, in 2013. No other has PI's name here because these are companies, but his lab was the one which identified that. And that's when he identified, and now see how many compounds are there. People have also made PROTAC degrader. Now they have also made the RAS in the on form inhibitor. Once you know, then you can target it. Okay, so look at this. How many compounds are there? Again, so many mutants there. Amgen, Merck, all the big companies are there. This is a huge deal, huge deal. Okay, look at that. Can you believe that? Such a recent discovery. Preclinical, these many. Phase 1, phase 2, phase 3, these are approved for RAS. Right? So now let's look at some case studies here. So this is, do you know what is pan-RAS? What is the meaning of pan-RAS? Pan-RAS means it inhibits all the 3 isoforms of that. Pan means all of them. So pan-RAS means HRAS, NRAS, and KRAS. So look at these things. So many drugs against KRAS. They target the inactive form. These compounds target the active form of RAS. Okay, and this is KRAS, this is HRAS, this mutation. Okay, so look at all the secondary mutations which have arrived, and all the inhibitors are there. Okay, so this is just to telling you all these different mutations and their relative percentage in different kinds of cancer. Okay. All right, again, this is an easy lecture to follow because it just tells you all different mutations and all the compounds which are out there. Okay, so 2013 was when he started. Before that, it was just, you know, no compound. And let's look at it. How many compounds are made there now? Okay, so 2013 is when discovery of the switch to binding pocket. This is the one, his discovery here. And then it is just so many compounds are there. These are target-wide KRAS inhibitors and their stage of development right now. Okay, timeline of case trial against various this thing. Okay, more compounds. Once you make the breakthrough discovery, then the world takes over. Okay, which is very good because we need all the things we can. Okay, so this is for KRAS, this mutation, G212 mutation. This compound is an orally active covalent inhibitor of KRAS for KRAS mutants. Okay, that's it. Not bad. 4 lectures finished.